Genetic origins of miscarriage. Risks begin before birth. Miscarriage is tragically common, affecting roughly 15% of clinically recognized pregnancies, though the true rate is likely higher, as many losses occur before detection. New research now points to a surprising reality. The roots of some miscarriages can be traced back to genetic variations present before a mother is even born. These variations impact how accurately chromosomes divide during egg development, increasing the risk of aneuploidy, a key cause of pregnancy loss. The role of chromosomal abnormalities around half of first and second trimester miscarriages are linked to embryos with the wrong number of chromosomes. This isn't random. It's often a consequence of underlying genetic predispositions. For years, scientists have known that maternal age increases this risk, but the deeper genetic factors have been harder to pinpoint. A massive genetic study reveals key links. A recent study at Johns Hopkins University analyzed genetic data from nearly 140,000 IVF embryos, providing unprecedented clarity. Researchers identified clear associations between specific genetic variations in mothers and an increased risk of aneuploidy. This is the clearest evidence to date of how genetic risk for chromosomal errors develops in humans. The key lies in genes controlling meiosis, the process where chromosomes pair, recombine, and separate during egg cell formation. Variations in genes like SMC1B, which holds chromosomes together during division, were directly linked to increased rates of aneuploidy. Other genes involved in chromosome crossover, such as C14orf39, CCNB1IP1, and RNF212, also showed strong associations. Why this happens? Meiosis and early development. Female meiosis begins during fetal development, pausing for years before resuming at ovulation. Genetic variations can disrupt this early process, weakening chromosome cohesion and setting the stage for errors when meiosis resumes later in life. The study's findings align with decades of research on model organisms, mice, worms, confirming the critical role of these genes in chromosome stability. What this means for the future while predicting individual miscarriage risk remains complex. Maternal age and environmental factors also play a role. This research unlocks new possibilities. Understanding these genetic roots could lead to drug development targeting meiotic instability and provide a foundation for studying paternal genetic contributions to pregnancy loss. The findings also underscore the power of large-scale genetic studies. Small effects only become detectable with massive datasets. Ultimately, this work highlights the deep biological realities of early human development and offers a new lens through which to understand the heartbreaking reality of miscarriage. This is not just about fertility care. It's about the fundamental processes that shape human reproduction.